And Alex? Oh, I heard it. Wow. Great, then we can start. Do you know what we record today? Great. That's how it's intended. Welcome to Easy Immunology. My name is Stefan, and in today's video we want to look at a immunological topic that is very relevant also for the clinical practice and millions of people out there. And I'm not going to tell you the topic at the moment because I want to work my way into it with my two colleagues. So as you might have realized if you have watched one of our videos before, I never do this alone. I have again with me today Alex. Hi, Alex.
Hi, Stefan.
And Hannah. Hi, Hannah.
Hi, Stefan.
These two are both masters in biology, and they help me by asking the questions you might have while watching this video, which has proven very helpful, and they also pay attention that I don't talk too much. So we will jump right into the topic with a clinical case presentation that comes out of my imagination, but it is based more or less in reality. So this is something very typical that you might see as a doctor. So we have a male patient that is 40 years old. He has two children, two small children, and he's always tired. And it doesn't even get better when he takes some rest. This is called fatigue. As a parent myself, I can tell you this could be caused by the two children, but here it isn't. He has also lost his appetite and lost already 14 pounds of weight. And you see he's not a big guy, so losing 14 pounds without even trying is something that is not very good. He also has frequent infections, and he has something like this. Maybe one of you two have an idea what this could be. Yeah, Hannah?
To me, this looks like a swollen lymph node.
Yeah, that's absolutely correct. So lymph nodes are secondary lymphoid tissues. We've made a video about it, and they can increase quite massively in size when there is an infection going on, because this is the place where the immune cells meet to start immune responses. So he has had this. He also has pain in his chest, so there's also something wrong. And he has a cough that doesn't go away, so he's coughing all the time. And he is easily bruised, and he has also this kind of skin symptoms. So this is like small bleedings under the skin, which we call petechiae. He has fever, and he is also sweating at night. And I've made the weight loss, the fever, and the night sweats bold, because that's what the doctor calls a B symptomatic, which is always a sign for a disease that is progressively getting worse. So something is not right with our patient, and it is rather serious, I would say. So maybe I can ask the two of them if they have any idea what could be going on with our patient. Yeah, Alex?
It's seems like there's a chronic disease going on. And you said it already, it's probably really severe, so it could be a tumor.
Mm-hmm. How did you get to that conclusion?
Because when a tumor is growing, the whole body is working on that, and it costs a lot of energy for the body to either fight or try to fight the tumor, or the tumor is also growing, and this is also costs a lot of energy, and this is causing the weight loss.
Hannah?
Yes, and the swollen lymph nodes is a sign that the immune system is somehow involved. So the lymph nodes are the places where the immune cells interact with each other and present information to each other. So if it's swollen, it means, okay, we have lots of immune cells in there. So this is also a hint that somehow the immune system is involved. And yeah, if it's a tumor, or it can be some kind of other autoimmune disease, I don't know. But yeah, this is, I think, Alex, you're right.
Yeah, so Alex was right. It is a tumor. It's a lymphoma. It's a tumorous disease where a part of the immune system is not working correctly. And we will look at what this means today. So today we will talk about tumor immunology. So tumorous diseases, cancer, tumor immunology are big fields. So we could fill a whole lecture series about the whole topic. But today we want to focus on giving a rather broad overview what a tumor is, how it arises, and then how we can use immunology to treat tumors, because there's a revolution going on in tumor treatment at the moment. And this is mostly caused by our better understanding of the immune system. So we will start, it's rather morbid, I know, with talking about how relevant the topic actually is. So I have looked into the database in Germany, what people die from, and I have ranked these causes for death in Germany, and I would ask the two of them if they have any idea what could be the top 3 causes of death in Germany. Yeah, Hannah?
Some kind of heart disease.
Mm-hmm.
I would also say smoking is probably quite good.
Mm-hmm. Anything else? Okay. It's not a problem. We will just look at it. So here are the top 3 causes, and also the difference between 2021, this was the most up-to-date numbers I could find, and 2011, so 10 years before. You can see number 1, undisputed, is ischemic heart disease. So this is exactly what Hannah and Alex were saying. So people die from their heart not working. And ischemic means that the heart is undersupplied, or any organ is undersupplied with oxygen. And this can happen from not training enough, from just being old, from smoking and destroying your lungs. So this is number 1. Then we have Alzheimer's disease, a disease that affects the brain and destroys the neurons in there. And we have COVID-19. Yeah, this is of course something that didn't exist in 2011. Now it's number 3 already. And if you look at the other diseases, there are things like stroke. So this is a clogging of a blood vessel in the brain that causes massive damage in the brain. We have COPD. It's a inherited disease where your lung doesn't work correctly. You build a lot of very thick mucus, and this is like a petri dish for bacteria. These people have frequent infections in the lung and die from it. Then you have chronic kidney disease, hypertensive heart disease. So this means having high blood pressure. This comes from not moving enough, from just watching TV and eating all the unhealthy foods we have. And then we have falls. So people die because they fall. Usually they are pretty old at that point, and then falling is the beginning of the end. So you should avoid this. So it's basically a sign for our societies becoming older. And you see, yes, cancer is among the top 10 causes, but it's not like in the first places. So this is good, and it shows how far we have come in treating cancer. So as you will see today, there is quite a bit of progress being made in treating cancers immunologically, and this will mean in future, probably very likely, that cancer will be like a chronic disease that you can live with for most of your life. Ideally, yeah. What you can also see, what I find quite fascinating as an immunologist, that there is just one infectious disease on there, COVID-19. So we have also made tremendous progress in preventing death from infectious diseases. So let's dive into the topic, and I will start with 2 definitions, cancer and tumor. So cancer is a disease that is caused by an uncontrolled division of abnormal cells in a part of your body or in your whole body. So we will look at in the next slides where this comes from, how this can be. And a tumor is a swelling of a part of the body that is generally without inflammation. So you can also have atypical inflammation. We have looked at this in previous videos, but this is usually with inflammation, so activation of the immune system, whereas A typical tumor is without inflammation. We will see today also why this is the case, because tumors can regulate the immune system. And this is again caused by an abnormal growth of tissue, and it can be benign or malignant. So benign means it's just growing in one place and doesn't do too much damage, and malignant means it causes quite a bit of damage, and it also spreads in your body and will eventually kill you. So, how does cancer arise? Cancer arises from normal cells turning into cancer cells, and this is triggered by certain factors, extrinsic factors, for example, like carcinogens. These are substances that cause this change from normal to cancer cells. Then there is genetic mutation. This is an intrinsic cause, which can again be triggered by, for example, carcinogens. So something in the DNA of the respective cell is changing. Then we have sun and other types of radiation. So a good part of cancer that we have in our society is caused by not using enough sunscreen, and this causes then damage in your genes. And also viruses and some other infections can cause damage to your cells and turn them into cancer cells. And what happens actually on a molecular level is that so-called proto-oncogenes turn into oncogenes. So an oncogene is a gene by definition that is changed in these cancer cells. And there are many different oncogenes, and the proto-oncogenes are the normal versions of these genes that fulfill their normal function. And these proto-oncogenes are usually genes that are involved in regulating how the cell grows, how it proliferates, so how it turns into identical daughter cells, and how the cell goes into apoptosis, so how it decides that, okay, my life is over now, I'm not useful anymore, I will just die in a controlled way. This is what apoptosis is. And examples are maybe transcription factors, so proteins that go into your nucleus and turn on and off a lot of different genes. Then we have GTPases. These are factors in the cells that regulate how the intricate network in the cell works and how these proteins and all the other things interact with each other. And then we have receptors, for example, for growth factors that signal via binding to the growth factor into the respective cell that it should grow and divide. And if this system is somehow damaged or changed, it can lead to normal cells turning into cancer cells. So this growth of cells and how cells divide is controlled by something that we call the cell cycle. And this is how a typical cell cycle looks like. You might have seen this in high school. And it is divided into different phases. So normal cells in your body that fulfill their normal function, like for example liver cells producing liver enzymes, they are in something that we call the G0 phase. So they fulfill their normal function. And then if the cell decides, okay, I need to divide, I need to make 2 or more cells out of myself, they leave this G0 phase and they then prepare their genomic material for the division and all the other stuff in the cell. Because basically if you have one cell and then you want to turn into 2 identical daughter cells, everything in yourself needs to be duplicated and divided onto the 2 daughter cells. Then it starts in the so-called S phase. This is the synthesis phase. There the DNA is replicated. Before the S phase, your chromosomes contain of 1 DNA double strand, and after the S phase, the strand was copied into 2 identical strands that are held together by a structure that we call a centromere. Then the cells get some rest because this is an intensive and energy-consuming process, and then they turn into the M phase. This is the mitosis phase. There first, all the chromosomes are lined up next to each other in the cell. It's fascinating. Certain spindles attach to the centromeres, and then they pull apart both parts of the chromosome, which are called chromatids. And then the cell divides in the middle, and we have 2 small identical cells where the chromosomes again consist of just one chromatid. Then the cell goes into G1 phase where it recovers a little bit, and then it can decide, okay, I can either switch to the G0 phase and fulfill my normal function, or I can grow again. That's why the lower cell is a bit bigger. Take some rest, recreate myself while preparing for another round of division. So this is the normal cell cycle. And this results in one cell being able to turn into many identical daughter cells. And this process, the cell cycle, is controlled by something that we call checkpoints. So there are 3 checkpoints in the cell cycle. The first one is between the G1 and the G0 phase. Here the cell needs to decide, do I need to divide myself, or do I just go into the G0 phase, or do I stay in the G0 phase and fulfill my normal function? Then there is the second checkpoint. It's at the end of the G2 phase. And here the cell has gone through the S phase and the G2 phase. And the cell then checks at the end of the G2 phase, did the duplication of the DNA and all the components in the cell work? Am I really ready to go into the mitosis phase? And then we have the last checkpoint that is at the end of, or towards the end of the M phase. And there the cell checks, okay, did the lining up of the chromosome works, and are all the spindles attached to the centromeres, and can I really start pulling everything apart and turning into 2 cells? Because once you start, there is no way back. Yeah, you will not get this put together. So these checkpoints make sure that this really complicated process works. And if now one of the many proteins that control the cell cycle has a malfunction, this cell will be damaged, and it might turn into a cancer cell. And in these cancer cells, all checkpoints are, if you imagine this as a traffic light, they are all put on green. So no matter if the cell is ready or not, or no matter if there have been mutations accumulated, the cell would just go through the cell cycle non-stop and will produce a lot of these damaged cancer cells. And even if it starts with a single mutation, these cells are not checked anymore. They divide uncontrollably all the time, and this leads to even more mutations. So the genetic stability that is more or less guaranteed, or should be guaranteed, but the cell cycle is not given anymore. And that's why cancer cells change quite dramatically genetically over time. So we now understand that this is happening by a deregulated cell cycle, and if this growth of cells goes on for long enough, they might form a tumor. So they will then form like a structure, an organized, more or less organized structure in your body that can be seen, can be felt, and can be found in diagnostic procedures that we call a tumor. I've brought an example of a cancerous disease, and this is called the Philadelphia chromosome. This is a specific abnormality in patients that have a disease that is called chronic myeloid leukemia, CML. It's a disease where cells of your immune system divide uncontrollably, and you can see this in diagnostic procedures by looking at the chromosomes. So there is something that you call a karyogram, where you somehow fascinatingly can isolate the chromosomes, you can stain them, and you can look at them under the microscope, and then you find neatly arranged pairs of chromosomes, because you usually have 2 of all the chromosomes except for your X and Y chromosome. And you can see that in patients that have this disease, chromosome 9 and 22 look a bit different, because as you see in chromosome 9 on the left side, one of the strands is a bit shorter, and in chromosome 22 on the left side, this strand is a little bit longer. And what happens here is you have here the normal chromosome 9 and 22, and there is a chromosome break and a reshuffling of the chromosome material, and part of chromosome 9 gets then attached to 22 and the other way around. And this fascinatingly always happens in the same place. And you get then this altered chromosome 22, where two genes are fused together. One is called the BCR gene, the B cell receptor gene. It's part of the B cells, very important cells in the adaptive immune system. And the other one is the ABL gene, the Abl kinase. And the Abl kinase is one of these molecules that regulates the cell cycle. And this means that in these patients, in B cells, because these are the only cells that express the B cell receptor, this cell cycle is disrupted. And then the B cells divide uncontrollably and cause a lot of damage in your body. And then you develop this chronic myeloid leukemia. As I said before, cancer can be, or tumor cells can be benign or malignant. Here I brought you two examples. One's called an adenoma, that is a benign change in some cells that make the epithelium of some glands in your body. And they just grow in one place, they form a tumor. But this tumor is happy to be there. Maybe it doesn't even grow so fast and doesn't cause any damage. And then you have malignant types of cancers like adenocarcinomas, and these might form a tumor, but the cells also change. You see, they look different. They are pretty degenerated compared to the cells that they arise from. And these cells also wander around in your body, and they cause a lot of damage because they will spread in your body uncontrollably, and they will form new tumors in all your body. And this is what we call a metastatic behavior, so that some cancerous cells spread from one body to unconnected parts of your body, and then you have not just one tumor that you can maybe cut out in a surgical procedure, but then you have small tumors everywhere. And in certain organs like the brain, it's very difficult to cut something out without causing too much additional damage. So this is then very difficult to treat, and this is a big problem with these cancer diseases, that you need to basically find them very early. When it's just one small tumor, then you have a good chance to control it, but once the tumor has spread, it gets much more difficult. I would like to spend one slide on the classification of tumors. I know this can be much more complicated, but for a general first overview, I think this is enough and this is good. So a cancerous tissue, so some part of your body that has turned into a cancer or a tumor, is called a melanoma, and they can be named either by the affected organ. You might have heard this, XY has lung cancer or brain tumor or kidney cancer. Or if you want to be more specific in a medical sense, you can group the tumors according to the type of degenerated tissue. And there are solid tumors where you can really find a tumor, an accumulation of tumor cells in your body. And they can be further subdivided into 4 types. So we have blastomas. They arise from mutations in cells that form your tissues and your organs. So these are the stem cells, and this is what we often see in children, unfortunately, that these cells degenerate. And I put it red here because this is usually a big problem, yeah? So cancer in children is not very frequent, but when it is there, it's usually not so good, because these tissues have a very high potency to regenerate and to multiply. And then if this happens, we have very aggressive and very fast growing tumors that cannot be controlled usually. Then we have carcinoma, so where epithelial tissue turns into cancer cells. This is also often malignant, and so the epithelial tissue is like the if you want to make it very, very simple, is the upper layer of an organ or of a blood vessel or something. And if this turns into cancer, we call this a carcinoma, and this is often very malignant. Then we have sarcomas. There, the supportive or connective tissue or fat tissue, something like this, turns into a tumor. This is rarely malignant, so this is good. This can be treated quite quite good, or maybe it doesn't even need to be treated. And then we have teratoma, where pluripotent stem cells like germ cells turn into a tumor. And this is also rarely malignant. And then we have systemic tumors, a systemic cancer, where you don't necessarily find a solid tumor, so something you can feel and touch and can excise, but certain cells turn into tumor cells and they spread in your whole body. And there we have two types. You have leukemia, where cells of the hematopoietic system, so cells that form, for example, your blood, or cells of the innate immune system turn into cancer cells. And then we have lymphoma, where it's the lymphocytes, the T and the B cells, that turn into cancer cells. And this is what we observed in our clinical example at the beginning, and also in the Philadelphia chromosome. There it was the B cells. Okay, this is just some classification of tumors. And now I'd like to go a little bit into tumor biology with you. And this can be studied in mouse models. I know it's not the nicest thing to do, but this is what we can do, where we can establish models of these complex diseases. And what you can do is you can experimentally induce tumors by exposing the animals to chemicals, carcinogens, irradiation, so triggers that turn these proto-oncogenes into oncogenes. Then the mouse will develop a tumor, and this tumor, or the cells in this tumor, will grow uncontrolled and turn into a bigger tumor, and then will kill a cell. Yes, Hannah?
Why is the mouse pink?
You are too smart. The mouse is pink because I want to make a point out of it in one of the next slides. But it's good that you asked. The mouse is pink because it doesn't have an immune system, and there are certain models of mice that do not have an immune system. They do not have hair. They are naked mice, and that's why I made these mice pink. And most of the initial work on cancer cells in mice was done on these animals, because as we will learn in a few slides, usually your immune system is very good at eliminating tumors in very early stages. And therefore, if you want to have a chance to reproducibly grow these tumors or induce these tumors in mice, you need to use either very aggressive tumors or mice that do not have an immune system. Okay? You can also take live tumor cells and inject them into the mouse. It will form a tumor that will kill the mouse, and you can then isolate some cells from that tumor again and transfer them into the next mouse, and it will develop the same tumor. So once the tumor cells are generated, induced, they can be propagated more or less forever. However, if you destroy the tumor cells that you transfer, then the mouse will not develop a tumor and will live a normal and happy life. So this means the tumor cells have to be alive. It's not just the trigger of the tumor that causes this response, but the cells have to really be alive and have to divide. So now we come to Hannah's question. The tumor is recognized by the immune system, and there are certain molecules on the surface of all of our cells, so most of our cells, that are called MHC molecules, major histocompatibility complex proteins. And they are, as we will also learn in the lecture about transplant rejection, they are the major antigens that drive recognition of cells as foreign or damaged or dangerous. And if these MHC molecules are different between the tumor and the cells of the mouse, then the immune system of the mouse will recognize the tumor as foreign and will be eliminating the tumor. But if the tumor cells have the same MHC molecules as the mouse, then the mouse will, or the immune system of the mouse will usually accept the tumor, and the tumor can grow. And the first, so the left part, is usually the case in also our clinical patients, because the tumor cells arise from our own cells, and they have the same MHC molecules on their surface as all the other cells in your body, and therefore also usually the tumor cells have the same MHC molecules, which makes it way, way harder for the immune system to recognize them as dangerous. Finally, you can also use an immunocompetent mouse, so it has an immune system, it has hair, that's why it's black and not pink. And then the immune system of the mouse will usually recognize the tumor cells and will eliminate them before they can form a tumor. So if the immune system is now so good at identifying tumor cells and killing them, why do some patients and a quite big portion of our population then develop tumors over time? And this is explained by the so-called 3E model. And I would like to explain this to you now. So imagine you have a cell in your body that has one of these mutations and turns into a cancer cell. These cells usually are somehow different than the cells in your body, and they maybe express some molecules, overexpress some molecules. We will look at this in more detail in one of the next slides, which allow, for example, NK cells to recognize them as foreign, and they will kill them. And also CD8 positive cytotoxic T cells, T killer cells, can recognize them together with the help of antigen presenting cells and CD4 T cells, and kill these cells at early stages of tumor formation or cancer development. And this happens in all our bodies all the time. So there are always a few cells that have a damage in their genes, and they are on the way to turning these cancer cells, and they are found and eliminated all the time in your body, so that you do not develop cancer all the time. However, by the action of the immune system, so the killing of the degenerated cells by the immune system puts pressure on them. So they want to survive maybe, and therefore, as I said, they change all the time because the genetic stability is not guaranteed anymore due to these changes in the cell cycle. And these cells then accumulate additional mutations that maybe allow them to grow even faster, to maybe wander around, or maybe stop the production of the molecules that allows the immune system to recognize them as foreign and dangerous. And then there's always some cells that change, and the immune system then maybe finds another way to kill them. And this is then the second phase. This is called the equilibrium, where tumor variants arise that are more resistant to killing, but still the immune system can somehow control this tumor. And then there is the one or two very important mutations for the tumor that allow then the tumor to escape the immune system by either controlling the immune system. You see here, this tumor has induced this gray T cell, it's a regulatory T cell, that can then suppress the immune system. So this takes off the pressure. And also you see some of the tumor cells have maybe accumulated mutations that allow them to wander away to other parts in your body, maybe your brain, where they are more protected from the immune system. And this is then the escape where the tumor or the tumor cells can spread more or less unchallenged. And these are the 3 E's. Yeah, the first is the elimination, which happens in all of us. Then there is the equilibrium, where there is a constant fight, but none of the sides is winning. And then at the end, there is the escape, where the tumor has taken over. Unfortunately, this is something we see all the time in patients that are immunosuppressed, where the immune system is suppressed. You can go from elimination, equilibrium to escape rather fast. So we see this frequently in patients that get an organ transplant because they maybe have a kidney disease. They get a new kidney, and in order to prevent the rejection of the kidney, they take strong immunosuppressants that the immune system doesn't destroy the new kidney. And then also this whole process is disrupted by suppressing the immune system. And then you go from maybe equilibrium to escape within a few weeks, and you develop a tumor that was controlled before rather efficiently. Yeah. So this is the 3E model, a very important model in tumor biology. One quick slide to how cancer is diagnosed. So imagine you have a patient, female one, with a breast cancer, and you want to learn more about the tumor and how to fight it. So the first thing you do is you take a biopsy. So you take a small sample from the tumor and you look at these cells, because as you can probably imagine, every tumor in every patient is somehow different. So everybody has different mutations, and every tumor has different properties, and it makes it like an individualized thing to treat the tumor efficiently. And then two things are done. The first one is called histology, and there you take the sample or part of the sample, you extract it from the patient, and you look at it under the microscope. And if you are an expert and histologist, you do this all the time, and you can see how far the cells have come from turning from normal body cells into these cancer cells. And here you see my crude depiction of different states of degeneration, and you can then already judge, okay, this is still very close to the original situation, or this is very far ahead and probably has also spread to other parts of your body. Then you also take some DNA from your cells and you do genetic analysis, and there you look which proto-oncogenes have turned into oncogenes. And this tells you a lot about the properties of the tumor. So what does it need to grow? How fast will it grow? How likely it is to form, for example, metastases. So there a lot of information is known. And if you put these informations together, you know, okay, this is a tumor that is pretty far along in his journey of degeneration, and maybe this over expresses a receptor called HER2. This is a receptor for certain hormones, which is very typical in breast cancer. And then you know, okay, this is a HER2 positive breast cancer. And then you know, okay, maybe I have to treat this one by blocking the HER2 receptor. And then you have certain drugs that block the HER2 receptor, and then you can treat this kind of tumor pretty efficiently. Whereas the tumor is, if the tumor is a breast cancer that doesn't HER2, you can treat the tumor all day long with these drugs against HER2 and it will have no effect. So this is what we call a stratified therapy, because we treat the tumor according to what it is and not essentially to where it was found. So if you have like a metastasis of a breast cancer in your colon, it might still be HER2 positive. So we need to treat it as the breast cancer that it once was, but not as a colon cancer maybe. Okay, and this can be done, as I said, with antibodies, and the most famous one is called trastuzumab. Okay, now after giving this general overview about tumor biology, I know I said in the beginning it's a rather broad overview, we will turn to how the immune system usually recognizes tumors and how we can use the immune system to control and treat these cancerous diseases. And the most important problem we have, and that you need to understand, is that the tumor is mostly self-tissue. So the tumor once was a normal cell in your body, and as we have learned in one of the previous lectures, there is something that we call tolerance development. So your immune system learns what is normal in your body, and it learns to basically ignore all the normal cells in your body, because if it wouldn't, it would kill you all the time. This is what happens in autoimmune diseases. Your immune system recognizes your own cells and tissues as foreign and dangerous, and then it starts to destroy these tissues. And if these tissues are important for you, then you will get sick and maybe die. And the same is true for tumors. They are mostly self-tissue. They shouldn't be eliminated by the immune system. But now these cells have changed, but maybe not enough to be recognized as foreign and dangerous. And this is a big problem. So how does the immune system normally recognize tumors and cancerous cells? So here, for example, you have a tumor that is growing, and some cells will always die during this process, will be damaged, and then parts that are normally in the cells also come out. And these can be recognized by so-called antigen-presenting cells, and they recognize tumors, for example, via these tumor antigens that are released from the tumor cells, and they bind to antibodies, IgG antibodies that are recognized via so-called Fc gamma receptors. These are antibodies that act as adapter molecules, then the cells take up and they maybe induce immune responses by presenting tumor antigens. Tumors can also be recognized by scavenger receptors or C-type lectin receptors. These are pattern recognition receptors that usually recognize pathogens by structures that do not exist on our own cells. And for example, as we will see in one of the next slides, certain sugar structures are overexpressed on tumor cells that we would normally find on, for example, bacteria or fungi, and this can allow the immune system to recognize these cells as dangerous. Then there can also be direct interactions, so the antigen-presenting cell scans the surface of the cancer cells, and there it can find antibodies that might have bound, it can find complement proteins, and it can recognize these carbohydrates also directly on the surface of the cell. And if this happens and an immune response is initiated, the tumor would be eliminated. But if this system does not work, the tumor would escape. This is one of the E's in the 3E model. However, if this recognition works, then usually antigen-presenting cells will induce naive T cells. They turn into effector cells that activate B cells. And together, these cells usually destroy the tumor. So this is what we call immunological surveillance. This is an example, just an example. You can directly forget it. There are many different types of C-type lectin receptors on the surface of the antigen-presenting cell that recognize different sugar structures. Also, I do not know most of them, but the ones that I put in bold letters are the ones that are overexpressed on certain tumors, whereas the ones that are put in gray are usually structures that we find on pathogens. You see, there are very specific carbohydrate motifs that can be changed in these tumor cells, which allow then the cells to recognize the tumors. And you see there are also different types of antigen-presenting cells. Ones are called conventional dendritic cells. This is the gray part, and some are called plasmacytoid dendritic cells. These are the pink ones, and they differ, at least in part, in their expression of the C-type lectin receptors. Okay, besides the expression of these danger molecules that we normally find on pathogens, the tumors also release some proteins, some factors that have usually nothing to do with pathogens, but they can still activate the immune system. And these are called DAMPs, damage-associated molecular patterns. And some examples are the high mobility group protein B1, some heat shock proteins, some ATP that is normally inside the cells, or uric acid that is normally inside the cells. But once the cells are damaged, this stuff comes out and can be recognized, triggering again an activation of T cells. And these DAMPs are also called alarmins because they alert the immune system towards a point of damage, point of tumor development, maybe also a wound that you have where these molecules are then released from the cell. And this can then initiate and drive, meaning perpetuate, non-infectious inflammatory diseases. So there is no pathogen around, therefore it's non-infectious, but still you find a immune response that is initiated. If we now look at the induction of immune responses by the tumor, you can see that the activation of the T cells by these antigen-presenting cells requires the presentation of tumor-derived peptides, so small fragments of tumor-derived proteins, to the T cell receptor via the MHC molecules of the antigen-presenting cells. And this, in end effect, determines the specificity of the tumor cells. So they will only be activated if the T cell receptor matches that peptide, and the other way around, only T cells that recognize these tumor peptides will be activated, but not all the other, for example, pathogen-specific T cells. Let's look at these antigens, there are different classes that I want to go through with you, and they are called tumor rejection antigens. So these are the antigens that lead to the induction of T cells, and the T cells then destroy the tumor. That's why they're called tumor rejection antigens. And there are different classes. The first class are so-called neoepitopes. So neo means new, and epitope is that the part of the protein that is presented. And these are new versions of normal proteins that only exist if you have a tumor. And this is pretty easy to understand if you think about the first example I brought, the Philadelphia chromosome with the BCR-able fusion protein. And then part of the BCR is fused to parts of this able protein, and then a new peptide will be generated. It's maybe half of the BCR protein and half of the able protein. And this is something that does not exist outside of tumor cells. So these antigens are strictly tumor specific, and therefore this is not part of the tolerance development. And then T cells can be activated that recognize this antigen. Then we have something that we call cancer testis antigens. These are proteins that are normally only expressed in male germ cells, so sperm cells, but they are maybe overexpressed in melanoma, so cancer of the skin or breast cancer. Then we have differentiation antigens. These are genes or proteins encoded by genes that are expressed normally only in certain types of tissues. Tyrosinase is an example. This is strongly overexpressed in skin cancer. Then we have an abnormal gene expression. So certain genes are strongly overexpressed, and the proteins are made in much higher numbers in tumor cells than in normal cells. And then these cells turn into cancer cells. And one prominent example is HER2/neu. This is then the example from the diagnosis I told you before. This is a receptor for a growth factor that is strongly overexpressed in these breast cancer cells, allowing these cells to bind more of the growth-inducing factor and to divide even stronger. Then you have proteins that are abnormally post-translationally modified. So translation is the process of turning an mRNA into a protein, and then there are certain modifications that happen after this, so post-translationally. So certain sugar molecules are added, for example, and these processes can be massively disrupted in cancer cells. And then you maybe have proteins that are strongly over glycosylated, so meaning they have much more of these sugar molecules. And then you can also have, for example, molecules that are not correctly glycosylated. And this happens, for example, in a mucin protein, a protein in your mucus. So this is the glibbery stuff in your lung, for example. And if this is strongly under glycosylated, it doesn't work. And then it can be recognized as foreign and dangerous. Then you have abnormal post-transcriptional modification. So transcription means putting your genetic information into an mRNA. And if there is, for example, additional information included by the retention of introns in the mRNA, this results in a change in protein sequence. And then there is additional information in there, or frame shifts happen that totally change the sequence, and then you have new peptides that can be generated from these proteins. And then there are, lastly, some few examples where, for example, viruses like the human papillomavirus cause a transformation of infected cells, and then they produce, for example, virus proteins that can be then overexpressed in the infected cells if they turn into cancer cells. So how are tumor cells normally surveillanced? And this is done by two types of cells, the NK cells and the CD8 T cells. And we will start with the NK cells. So imagine you have a normal cell, like a liver cell, is always my example, that produces liver enzymes. And this cell, like most of your cells in your body, then shows on its surface what it's working on its inside. So it makes a lot of liver enzyme proteins, and they are then also turned into peptides, so shorter fragments, and these fragments are then presented on the surface of the cell on the MHC class 1 molecules. So these cells continuously show on their surface what they work on in their inside. If the cell now turns into a cancer cell, it might be stressed because it realizes, oh, something is not right with me. And then it sends stress signals that can activate the NK cells, and they can then kill the cell that is on the way to turning into a tumor by secreting molecules like perforin that punches holes into the membrane of the cell, and then proteases and defensins go into the cell and induce a program that we call apoptosis. So this is controlled cell death, where the cell basically kills itself in a clean way that it can be recycled. This action of the NK cells can also then be enforced by cytokines, so small molecules that transmit information, for example, by macrophages. But if the cells are further along in their journey to becoming a cancer cell, they can also shut off the presentation of peptides. And then they cannot be recognized as dangerous anymore, because the NK cell just does not realize them anymore. But still they are killed, yeah. And this is what we call the missing self theory that was awarded a Nobel Prize to Professor Zinkernagel in the 90s. And this works because the system is a bit more complicated. So here you have again a normal cell that shows these, for example, liver peptides on its surface. And if the NK cell scans the cell, it gets basically two signals. The first one is it recognizes the liver peptides on the MHC molecules, and then it knows, for some crazy reason that is too complicated to explain at this point, that this cell presents liver peptides, so it is a liver cell, so everything is in order with that cell. So this transmits an inhibitory signal into the NK cell. Basically, the normal cell tells the NK cell, everything is right with me, please don't kill me. And there is also an activating ligand on the normal cell that is recognized by an activating receptor on the NK cell. So basically, it's a bit crazy of the normal cell. It says on the one side, everything is all right with me, please don't kill me. On the other side, it says something is not right with me, please kill me. Yeah. And if these signals are both there, both the inhibitory receptor and the activating receptor are engaged on the NK cell is somehow confused and does nothing. So the cell can survive. But if either the inhibitory signal is gone, so the cell has stopped expressing these MHC molecules, or the activating ligand is gone, so that's the right example. So both signals are gone. It doesn't matter if only the inhibitory one or the activating one is gone. The end result will always be that the inhibitory receptor is not engaged, and the cell will be killed. So this is how NK cells can then react to quite a broad range of mutations in these cells if they change towards becoming cancer cells. So if we look at cancer cells, there is a spectrum of these cells in terms of expression of a lot of molecules, and we will just look at MHC molecules. So there are cells that express high amounts of MHC, normal amounts of MHC, low amounts of MHC, or maybe no MHC at all. And the immune system has come up with a very clever system. So the activation of NK cells and the killing of the tumor cells by NK cells depends on the level of MHC expression. And the lower the MHC expression is, the lower the signal is that is transmitted by the inhibitory receptor. So the lower the MHC expression, the more the cell, the NK cell, realizes that something is wrong with that cell, and it will kill the cell, and there's a certain threshold. And there are other cells which we will look at in a second, CD8 positive cytotoxic T cells that also can kill cancer cells, but there the system is the other way around. Yeah, these cells definitely need the signal via the MHC class 1 to the T cell receptor to recognize that, there's something wrong. They need to to recognize these tumor rejection antigens via the MHC and the T cell receptor. And here they need a certain level of peptide presentation via MHC. And then the more peptides are presented, the better the cell can kill the tumor. So there, the threshold is the other way around. Yeah, and there are two different thresholds that overlap in the middle, making sure that for every level of MHC expression, there is a cell that can recognize the cell as changed and dangerous and can try to kill it. So I think that's a very elegant, very elaborate system that prevents many patients from developing cancer. And this can be, the system is true, can be controlled pretty easily or relatively easily in experimental systems by, for example, transfecting cells with MHC molecules. Then you make them more easily be killed by CD8 T cells, or suppressing MHC expression in tumor lines, and then they are killed more easily by the NK cells. Alex had a question.
You just explained it.
Okay, that's what I'm here for. Okay, great. So this is the missing cell. Then cancer cells can also be killed by a mechanism that is called ADCC, antibody-dependent cell-mediated cytotoxicity. It's Stefan, what are you talking about? So cytotoxicity means the cell is killed. Toxic is toxic and killing, and cyto is the cell. And antibody means, so the triggering factor that causes the cell to be killed is an antibody. And cell-dependent means, in the end, the cell that is killed by another cell. And what this means is basically that, especially at later stages of tumor development, there are certain antigens on the surface of the tumor cell that are not expressed on normal cells. For example, different sugar structures. They are recognized by antibodies because the immune system has made a B cell response against these antigens. And then these antigens act as adapter molecules that allow the NK cell to recognize the cell as dangerous and kill it. And this is very, very important, as we will see in the last part of the lecture, because these antibodies have, in a way, revolutionized the treatment of cancerous diseases, because they can be made in very pure forms in very big amounts to treat patients. Then we have the recognition of these tumor rejection antigens, these peptides from proteins that are specific to tumors by the CD8 T cell. And they can then kill the T cell by releasing perforin, which again punches holes, proteases, and granzyme B, which induces, or together induce, apoptosis. So, as I said before, tumor cells can be killed by these CD8 positive T cells, because we also call them cytotoxic T lymphocytes. That's what CTL means. They do this by granzyme and perforin, but they also do it by a molecule that is called Fas. Fas is called the death ligand, and it binds to a Fas ligand. These are both trimeric proteins, so they consist of 3 identical proteins next to each other, and this transmits a very strong signal towards the tumor cell to go into apoptosis. Besides the CD8 positive cytotoxic T lymphocytes, CD8 T cells that directly kill the tumor, also we have other T cells in our body. These are called the CD4 positive T helper cells. They are also very critically important in controlling immune responses towards cancer, because they are the managers of the immune system that regulate all immune response and also activate the CD8 T cells. They help establishing memory responses, and in a certain capacity they can also directly kill cancer cells. They can secrete cytokines, so small molecules in the immune system, and one of them is called tumor necrosis factor alpha. There, you already have it in the name. It's a molecule that induces death in tumors. Yeah. So if this system is so perfect, how can it happen that the tumors escape the immune system. And there are different things that contribute to this. The first one is especially relevant in the initial phase of tumors when they are still small. The first one is a low immunogenicity. You see there is a tumor cell, it doesn't release anything, no peptides that can be taken up by the antigen-presenting cell, it doesn't trigger any pattern recognition receptors, so there is just nothing there for the immune system to work with. Then it can also happen that certain molecules are released from the tumor. They are taken up by the antigen-presenting cell, but still the tumor doesn't trigger pattern recognition receptors, or the damage-associated molecular patterns are not there, so the cell is not activated. And if the cell only presents peptides via MHC molecules, but no co-stimulatory molecules and no cytokines, the T cell that is specific for that antigen will be tolerized, so this will not result in T cell activation. And then also some tumors can change over time. You see, it was an orange tumor cell, it turns into a purple tumor cell. So this is what we call antigenic modulation, and this happens if the immune system then starts to recognize the cell and starts to apply pressure on them. Yes, Alex?
As tumor cells are cells from self, and the antigens are taken up by the dendritic cells and presented to T cells. The dendritic cells direct immune response against self proteins.
Mm-hmm.
Could it be that after some time there is the initiation of autoimmune reactions?
That is a fascinating question. I am not aware of it, actually. If this would happen, I don't know.
I also never heard about it, so
Yeah.
it just came into my mind.
Yeah. It's a relevant point, but I'm not aware of it. So somehow the immune system must control against it, or
Seems like
it, or
we need
otherwise,
to go into literature.
Yeah, but I think
check.
there are unfortunately there are so many patients around with cancer, and if a big portion of them would develop autoimmune diseases, we would have probably heard about it. So, yeah, in the end, you still have tolerance development going on all the time. It probably eliminates most of them. Yeah. Yeah, but
Okay.
Fascinating question.
Thank you.
Okay, this is rather easy to understand. Now it gets a little bit more complicated, but I'll do my very best. Because at some point the tumor is not just hiding, but it starts to fight against the immune system. So it starts to modulate the immune system. So you have a tumor cell in the middle that has certain molecules on its surface and certain molecules that it secretes. And these little bastards, sorry for the strong words, they then regulate the immune system. And there are different molecules. For example, IDO, that is an abbreviation for indole 2,3-deoxygenase. This is a molecule that inhibits tryptophan metabolism. Tryptophan is an essential amino acid, so the T cells and all the cells that divide very strongly need a lot of tryptophan. And this IDO then blocks tryptophan metabolism, and basically the T cells then starve, and then they can't start immune responses. Then you have TGF-beta, transforming growth factor beta. This is a cytokine that the immune system normally uses to shut down immune responses at the end of an immune response. And this is then also secreted by tumor cells. So basically they shut down the immune system. And via certain molecules, for example, antigen presentation via MHC and certain co-inhibitory molecules, the tumor cells can induce their own regulatory T cells. So these are cells also normally part of your repertoire in your immune system to shut down immune responses at the end of an infection or to prevent autoimmune diseases. And these then produce more IDO, more TGF-beta, and also interleukin-10, which inhibits the activation of the T cells. So I said the tumor can induce the differentiation of regulatory T cells via the expression of co-stimulatory molecules and co-inhibitory molecules. You see here a dendritic cell that activates a T cell, and you see that there is a lot of communication going on between the antigen-presenting cell, the dendritic cell, and the T cell. So in the middle you have the MHC with the peptide that gets recognized by the T cell receptor, and then whether the cell which is specific via its T cell receptor to that peptide is activated or not depends on the additional signals it gets. And this is done by the mixture of co-stimulatory molecules, which transmit an activating signal into the T cell, and co-inhibitory molecules, which tell the cell basically, no, I'm presenting a peptide here, but this is totally normal. Please don't start an immune response. It's an important control mechanism in your body to prevent the establishment of autoimmune diseases. And you see there are quite some examples, quite complicated, just that you have seen it here. And that you can see, for example, CD80 and CD86 are very important co-stimulatory molecules on the dendritic cell. And they can be recognized either via CD28, for example, on the T cell, that results in T cell activation, or CTLA4 on the T cell, then it results in inhibition. And the CTLA4 has a much higher affinity, so it binds much stronger to CD80 and CD86 than CD28. And this can result, for example, in that T cells are not activated if they are instructed, for example, by tumor cells to strongly upregulate CTLA4. Then, for example, a co-stimulator signal can no longer be recognized by that cell. Here you see a clinical example of a patient that has a gastric cancer, so cancer in the gastrointestinal system. And on the left side, you see a normal, healthy control. And on the right side, you see this gastric cancer. You see the tissue looks quite a bit different. And there are a lot of these blue cells in there, and these are regulatory T cells. So there's a strong accumulation of regulatory T cells in later stages of cancer. And these cells then suppress the immune system. They do this by secreting certain cytokines. I'm not going to go into all of the details of that slide. If you want to look at it, you can press pause. But the most important ones are TGF-beta, transforming growth factor beta, IL-10 and VEGF, vascular endothelial growth factor. And they can suppress, for example, T cell growth, T cell differentiation. They inhibit cytokine secretion. They induce energy in the T cells. So they suppress T cells being activated. They can inhibit antigen presentation, shift the type of immune response that is going on, downregulation of co-stimulatory molecules, and induction of more regulatory cells. So there's a massive potential in these cytokines that is used by the tumor to modulate the immune system. So besides the modulation of the immune system by the tumor, also some tumors form something that we call a tumor-induced privileged site. So do you see this tumor has formed its own barrier, its own house, its own castle made of collagen, and then the immune cells maybe cannot reach the tumor anymore. Then it can grow happily within this tumor-induced privileged site. Okay, so now that we understand how the immune system normally fights cancer and how the tumor evades recognition by the immune system, we want to look at, in the last part of the lecture, how we can use the immune system to fight cancer. And this is, as I said in the beginning, where the revolution in cancer treatment is going on at the moment. So, what is the aim in curing cancer or treating cancer? It requires that all malignant cells are either removed or destroyed from the patient. That's very easy. You can just irradiate the patient with a very high dose of irradiation, and then all the cancer cells will be dead. The problem is all the other cells will also be dead, yeah? So you have to do it without killing the patient under ideal circumstances. And this makes it very difficult. We could talk like 3, 4, 5, 6 lectures about tumor treatment, but to make it very simple and to be able to focus on the immunological part, I've made this slide. So the goal is always to reduce tumor load. So to get rid of much as much of the tumor as possible. And this is usually done by this cascade. So the first one is surgery. So if you find a tumor, try to cut out as much as possible. Sometimes that's rather easy. Sometimes it's more difficult, because if the tumor is at some site in your body that is massively important, or if it's near to some arteries, or if it's in your brain, then you can't cut out that much. If you've done so, however, then you try or start to treat the patient with radiotherapy or chemotherapy. So under ideal circumstances, you have cut out all of the tumor with the surgery, but you cannot be sure. So there could be single cells still around at the tumor site, or some maybe have spread already to other parts of your body. And you try then to eliminate them with either chemical substances that destroy cells that strongly divide. This is, for example, cancer cells, but also immune cells or cells in your stomach. This is what we call chemotherapy, or radiation therapy. So you apply radiation to kill the cells. This is either done on the whole body, usually not. Usually it's done, for example, towards the chest, if the patient has a breast tumor. And this can then be repeated and either done at the same time. So maybe give a localized radiotherapy towards the chest in patients with breast cancer, and then they get systemic chemotherapy, or just one or the other. There are millions of combinations. And this usually leads towards such a strong reduction of the tumor load that you cannot find the tumor cells anymore in the patient, but there might still be around a few cells that could then start new tumors. This is then where immunotherapy is initiated. And then we try to use the immune system by the strategies I want to tell you in the next slides, by killing the last few remaining tumor cells. So at this point, I would like to ask the two of them if they have any idea when these types of treatments were first performed. Do you have any idea? When could the first surgery have been done, the first radiotherapy, chemotherapy, immunotherapy? Yeah, Hannah?
So I would say surgery is fairly old.
Mm-hmm.
I think, I don't know, but I would guess like maybe even medieval times. I don't know.
Even before. So we have found
Okay.
mummies from the Egyptians that were treated with surgery. So it's before.
Oh, even Egyptian.
Yeah.
Okay, so before Christ.
Yeah, before Christ.
And
Radiotherapy?
immunotherapy would be very new.
Yeah,
like
that's very new.
Yeah.
Any idea
couple
about
of,
radio or
50
chemotherapy?
years. Somewhere in between.
Yeah, somewhere in between. Alex?
I would say in the... Now it's the English word for 18th Jahrhundert.
Yeah, 19th century.
No, it's 18th century.
18th
I would
century.
say.
Yeah,
Okay.
I mean,
18th, 19th century.
way closer to the immunotherapy than to the surgery.
Yeah.
Mm-hmm.
19th century? 18th
Yeah,
century?
something like this. So, as I said, surgery, we found evidence that it was done by the Egyptians on mummies. Radiotherapy was started in 1896. It's crazy. This is where Röntgen and colleagues were discovering the Röntgen radiation, and then you could go to your local party at your village, at your town, and you could get your hand irradiated by Röntgen apparatus and see the bones. And this was like a party trick where many people developed cancer from later on. And chemotherapy is something that was started during Second World War, and immunotherapy was started in the 1970s. So this is a rather new development, but you can see we are making progress. So what are the aims of immunotherapy? So you have a tumor and you want to get rid of the tumor, and you want to do so in immunotherapy when tumor load is low. You remember the last slide? By providing the patient with anti-tumor effectors immunologically. These can be antigen-presenting cells, these can be effector T cells, or this can be antibodies. And ideally, you would not give this from the outside, because this is difficult and expensive to make. But ideally, you would trigger the immune system of the patient to do this by his or herself. Yeah, so you want to stimulate the patient's own anti-tumor responses. There are 5 things that are done that I want to quickly go through with you. And the first thing are immune system modulators. And here the goal is to just give a boost to the immune system to start immune responses. So you want to break the tolerance of the immune system towards the tumor that the tumor has induced. And this is done by giving cytokines. So cytokines, as I said multiple times already, are small messenger molecules that transmit, in this case, an activating information to the immune cells in your body. And the 2 most famous ones are interleukin-2, IL-2, and interferon alpha. And they both promote the activation of T cells. They are used for metastatic, so cancer that has already spread, melanoma, so this is skin cancer, and renal cell carcinoma. This is like kidney cancer. They activate the immune system, but this is not without side effects. So this causes death in 2% of the patients. It's not so good. Yeah, it's a rather high rate of death. I would not like that as a side effect. And this is caused by something that we call capillary leak syndrome. So what happens is you inject huge amounts of these cytokines to trigger the immune system. That's exactly what they are doing. And they do this by changing, for example, the permeability of the cells in your blood vessels, and that immune cells can migrate over the blood vessels into the tissue, and then also a lot of fluid in other cells migrate into the tissue, and this can cause a lot of damage and loss of blood pressure and something like this. Interferon alpha induces MHC1 expression on tumor cells. You make them more killable by the NK cells and the CD8 T cells. And it enhances the activation of dendritic cells, macrophages, and also the NK cells and the T cells. And this is done then again for melanoma patient, a renal cancer patient, but it causes neuropsychiatric diseases and changes your brain, basically damages your brain because the cells are activated very strongly, and some of them obviously go into the brain and cause a little bit of damage there. And it then can also cause autoimmune diseases. Maybe this is a bit into the direction of Alex's question, and it can destroy your thyroid gland. Then you develop something that is called hypothyroidism. So your thyroid gland is damaged, and your metabolism basically tanks and goes down way stronger than it should, and it can also cause vitiligo. And this is where I brought you 2 clinical examples. So vitiligo is an autoimmune disease that destroys the melanocytes in your skin and in your hair. And the melanocytes are the cells that give your skin the color. You see on the left side, you maybe remember him. That's Michael Jackson, a very famous musician. On the right side is a famous model that I don't remember the name of because I'm not into models. But you see they have these white spots on their skin, and this is caused either by cancer treatment on the right side or by some also inherited disease, probably in Michael Jackson, nobody knows. And therefore these melanocytes are destroyed, the skin turns very white. This is only a cosmetic problem, so nothing is wrong with your skin, it's just white, but of course you can imagine this causes some problems for the affected persons. Then the second strategy is something that we call therapeutic vaccines, or maybe even prophylactic vaccines. And this was initiated by results, again, from mouse models, where you take a mouse, you immunize it with killed, irradiated tumor cells, and this mouse will not develop a tumor. But then if you take the same mouse and inject it with live tumor cells that would normally result in tumor formation, this mouse also doesn't develop a tumor. And this means, if you think about it, that this mouse, with the first application of the irradiated tumor cells, has made an immune response that can protect it from the live tumor cells. And if you, I know it's a mean model, then take the same mouse and give it a different tumor, not the one that was irradiated in the first place, then it will develop a a tumor. So this is a specific response that can be induced towards a certain tumor. Yes, Hannah?
Is this what they do with the cervical cancer vaccine?
Yes, because this is caused by human papillomaviruses, and if you immunize young children, both male and female, against these viruses, they will form immune responses, and then you will prevent that these viruses can infect them and cause the transformation of normal cells into tumor cells in the female child. Yes, Alex?
But then in theory, it would be possible to just create a vaccine that includes like the tumor markers we were talking before, like HER2, for example, and create vaccines against, or create one vaccine against many tumor markers. Would this be possible?
In theory, we will come to this in a point. This would be possible. The problem is, yes, HER2 is a very, very frequent antigen in certain tumors, but it's just one. And many tumor cells have developed multiple mutations. And then even if you treat one, maybe there are other factors that are changed in these cells, and you will see it's very hard to hit all of them, and every patient is different, and that makes it quite a bit different. That's also why this was done in a certain way, as I will tell you on the next slide.
And I can imagine that if you have no tumor, but you vaccinate against these kind of proteins, maybe then we have the autoimmune disease we were talking about before.
That is maybe a complicating problem, and also I think in our modern society, many people will not understand why they should be vaccinated against 100 different antigens that are self in a way.
Yeah.
But yeah, in theory, as you will see, this works. So how is this done? This is called adoptive T-cell therapy. So what you have is you have a patient with a tumor that gets treated with surgery and radiation. So again, we are in the immunotherapy stage where the tumor load should be pretty low. Then you isolate some T cells from the patient that are tumor specific. So there has been an immune response initiated, but it is suppressed by the tumor. Then you give strong stimuli that activate these T cells, like interleukin-2, from the first surgery, but you do it now outside of the body. And some anti-CD3 that activate the T cells, and you have some antigen-presenting cells, and then you give it back, and then you hope that these activated fight-ready T cells then attack the tumor. This can be done, but you have also the chance that you activate some non-tumor cells in there, and therefore, usually you take some tumor biopsy, which is taken anyway during the surgery, and you add then this tumor cell lysate and maybe some adjuvants that trigger an even stronger T cell activation that you rather specifically activate than the T cells that are really specific for the tumor. Yeah, this was done. There was one product on the market that took out the slide. It is a vaccine against prostate cancer. It worked. But it improved the survival of these prostate cancer patients by only 4 months. And it cost like 100,000 euros. That's why it's not available on the European market anymore, because the health insurance has to pay and it's not financially viable. In the US, it's still on the market because there you pay for your own treatment, and then people are maybe willing to pay this for the 4 months. So, yes, Hannah.
So basically there is the vaccine, like for example for the cervical cancer, that you give in advance. So it's
prophylactic.
So basically there's a prophylactic type of vaccine, like for the cervical cancer. Mm-hmm. And there is a vaccine that you give when the patient already has cancer.
Therapeutic.
Made from their own cells.
Yeah. So that's exactly true. Prophylactic you give before you even have the cancer, and then there's therapeutic, when the patient already has cancer. And this is, of course, much more difficult because then all the modulation has happened. The cancer is maybe already several months or years old and has done all kinds of damage in your body and in your immune system. The advantage here is you don't have to think about things like histocompatibility. So are the T cells compatible with the specific patients, because they always are in this setting because they come from that patient. Yeah, you take the patient's own cells and then give them back. But this also makes it more complicated because you have to do this for every patient. Yeah, that's why it was so expensive. You have to take patient blood, isolate the T cells in a rather sophisticated, very well controlled lab, activate them, and then ship them back and inject them into the patient. So that's why it is so complicated. Yeah. So in a way, these vaccines are designed to stimulate the immune system of the patient, but the problem is that the tumors are poorly immunogenic, so they don't want immune responses to be induced against them. And they are difficult to develop because, as we just discussed, there are many different antigens that vary from tumor to tumor. Tumor peptides can only be presented if you have the matching MHC molecules. You can produce all the tumor peptides all the time you want, but if they do not fit in your MHC molecules, then it doesn't help you because then the immune system can't be activated. And to make them effective, you must probably include a lot of different tumor antigens, and therefore this can only be used when the tumor burden is very low. What might work much better, there Alex and Hannah were too smart for my lecture structure, is something that we call prophylactic vaccination. So this is now worked on quite extensively, and I think here, for example, mRNA-based vaccines are a huge opportunity because there you can generate these different tumor antigens or the sequences of these tumor antigens very easily. So what you do is you vaccinate the patient, you induce an immune response against the tumor antigen, and he forms then these anti-tumor killer cells that do nothing as long as the tumor is not around. But then if the patient later on, after the vaccinations, can be months and years later, develops the tumor, it can be killed in really early stages by these anti-tumor cytotoxic T cells, and then you will never even realize that you were on the way to developing cancer. But as we discussed before, this has a lot of problems because there are many different tumor antigens that you need to be aware of, and there is the chance of inducing autoimmunity. The third strategy that from now on things will be very successful. So strategies 3, 4, and 5 is what's revolutionizing tumor therapy already at the moment are so-called therapeutic antibodies. This is a big group of drugs. Therapeutic antibodies are, in a way, drugs that are used to instruct the immune system to attack the tumor or to attack the cancer cells. The thing is, they then act as adapter molecules to activate the immune system, but they can only usually be used against tumor-specific antigens that are expressed on the surface of the tumor, because usually the antibody does not go inside the cell. So you need to have structures on the surface of the cancer cell that can be bound by the antibody to have its effector function. And there are different types. They are usually monoclonal antibodies. This means they are derived from one B cell that was turned into an immortal antibody producer. So it's a clone and it only produces one antibody. Therefore it's a monoclonal. So these are very defined molecules. Then you can modify these antibodies. You can either act a toxin that then kills the cell that binds the antigen, or you can add a source of radiation. So you can have like a small, let's call it a small radiation substance radiating small atomic reactor, if you want to call it like this, that then transmits radiation and kills the cell that is bound by an antibody. And then you can also improve these antibodies by just using antibody fragments. So the upper part of the antibody is called a Fab fragment, and they are much smaller, and therefore they can penetrate much deeper into the tumor tissue, and they can also be modified then with toxins or radiative substances. So how do they work? They can either directly kill the tumor when binding to the antigen, for example by the irradiation, or by the toxin. And the toxins are cleverly designed. They only are toxic if the cell takes them up. The antibody, then it gets activated inside the cell and the cell will die, whereas the ones with irradiation just kill everything that is around. Then you have antibodies inducing this antibody-dependent cell-mediated cytotoxicity. Here the antibody just acts as an adapter molecule, allowing the NK cells to recognize the cancer as foreign. And then you can also have the antibodies used as tools to trigger the complement system. And basically this results in poor inclusion in the tumor cells, and the tumor cells that will then bleed out and be destroyed. Then you have certain tumors that need to be vascularized. So they need need to form their own blood vessels so that they can be fed by the rest of the body. And then there are antibodies that can prevent this vascularization, like the anti-VEGFR receptor or the anti-integrin alpha beta 1, alpha 5 beta 1. They then basically help to starve the tumor. How do they work? They can further also inhibit the dimerization of receptors. For example, you remember the HER2 receptor in this breast cancer patient. It is a dimer, so it consists of two proteins that need to come together to form an active receptor. And if you block one of these molecules, it cannot associate with the other one, and therefore there is no functional receptor, and the cell can no longer react to the growth stimulus. Then you can block the ligand that bind to the receptor, and you can just cross-link the receptor and use a very strong activating signal that is so strong that the cell decides, oh, no, this is all looking a bit shady, I just go into apoptosis. There are different target molecules for these antibodies. Again, I will not go into detail. You can press pause. It's quite complicated, but there are antibodies against, for example, CD20. Then you can kill all the B cells. There are antibodies against glycoproteins. There are antibodies against these angiogenic molecules, so against processes that lead to the formation of blood vessels. You can inhibit growth or differentiation signaling, or you can target specific glycolipids and carbohydrates. Carbohydrates are sugars. Glycolipids are molecules consisting of sugar and fat-like structures, and then you can specifically kill or recognize tumor cells. Problems with these therapeutic antibodies is they are very good at killing tumor cells, but only sometimes the outer shell. Yeah, and then you have a lot of dead cells and debris, and the antibodies just do not get deep enough into the pulsating heart of the tumor, if you want to call it like this, and you only kill or damage the outer shell. This can be improved by using smaller antibody molecules, but if you have a big tumor, there are patients with tumors that are like this big, you can imagine it's very difficult for the antibodies to reach into the whole tumor mass. Then there is inefficient killing. You see there are two tumor cells where the antibody cross-links the receptor. It's the same thing happening on both cells. One gets so strongly activated that it goes into apoptosis, but the other one just doesn't react to the signal. So this is inefficient killing. And some tumors are swimming in a sea of their own debris, and then the antibodies are mopped up by soluble antigens so that they don't even reach the tumor or don't reach the tumor in sufficient amounts. Okay, these are the therapeutic antibodies. And now we come to the immune checkpoint modulators. So what does this refer to? This refers to what we learned in the beginning, that the tumor modulates the immune system by all these mechanisms. And to make it more simple, It's a rather complicated process. I've made this slide. So imagine you have a T cell that is specific for a tumor, and it has two choices. If it's a cytotoxic T cell, it can either kill the tumor, that is the lower part, or it can ignore the tumor and the tumor can continue with its growth. So that's the upper part. And this is controlled by additional signals that the T cell gets. So if the T cell gets instructed by an antigen presenting cell via presentation of tumor-derived peptides and co-stimulatory molecules, it can get an activating signal, then it will kill the tumor. But if it gets, for example, a deactivating signal via these co-inhibitory molecules, then it can be instructed by the tumor, for example, to ignore the tumor, and then the tumor will survive. And this is done by many different signals. The two most famous ones are on the T cell side, the CTLA4 and the PD1. PD is programmed death 1, and it binds to PDL1, programmed death ligand 1 or 2. And the CTLA4 is also a co-inhibitory molecule that binds to CD80 and CD86. And this way of the tumor suppressing the T cell response can be regulated by antibodies. And there are two very famous antibodies that are anti-PD1 and anti-CTLA4. These are antibodies that bind then to PD1 or CTLA4, and therefore they block these receptors, and then the tumor can no longer instruct the T cell to ignore the tumor. So you modulate how the tumor modulates the immune response. So basically you make the T cell more easy to activate, and then it will no longer ignore the tumor, but it will start killing the tumor. Yes, Alex?
But I can imagine a lot of side effects in this case, right?
There are.
Because you have the high inflammation of T cells, like undirected to the tumor, but directed to everything.
Yes. Yeah. Basically, that's exactly true. There is a threshold at which T cells are activated, and you now start by, or the tumor started that fight. Let's be real for a moment. The tumor increased the threshold, so it says no. We first start an immune response very late, and now you come in with the antibodies and you lower the threshold, making the T cells more easily again to be activated. And this causes a lot of problems. You lower the threshold for T cell activation, making T cells more easy activatable, but this also results in a lot of inflammation, because this is not specific to the anti-tumor T cell. This happens in all the T cells, and then you will form like autoimmune responses and inflammatory responses. This is exactly what's happening. So there are different antibodies available in the clinics. One is called ipilimumab. It targets CTLA4, and it works miraculously, one has to say. So here is a clinical example. On the left side, you see a patient or different patients with tumors, always there where you have the white arrows. There's a tumor. So you have one in the lung on top. Also a little bit further down in the lung, you have quite a few small tumors, and you also have one in the brain. So this, especially the one in the brain, would be very difficult to get out with surgery without doing too much damage. And then the same patient was treated with the anti-CTLA4 antibody, and you see also the big tumor on the top and the tumor in the brain, they are gone. Yeah, it's crazy. So you can treat patients with this antibody, and 20% of patients treated with these antibodies have survived advanced melanoma for more than 4 years, and they would have died with 100% certainty without the antibody. So, and there are data that some of these patients survive for 10 years. So you could define them as cured from their cancer. But as Alex already alluded to, you start to mingle with the immune system, and you get a very strong activation of the immune system, which you use to kill the tumor, but you also get damage in the rest of your body. And this is from the same patient. You can see the uppermost one is a biopsy of the skin, then of the gut, and of the liver. And you can see all these small blue points. They are lymphocytes, T or B cells, that have invaded the tissue looking for something to kill, and they cause a lot of inflammation. And this is what we call immune-related, because the immune system is activated, off-target because it targets something else than the tumor, and toxicity because it causes damage. Yeah, and this is of course something you might not feel very good with as a patient. Yeah, and you have to imagine that the patients that are treated with this, they have cancer, they are not the fittest, and this can be a problem. Yeah, so there are these severe life-threatening side effects like global skin inflammation, gastrointestinal inflammation, damage to the kidney and the liver, and this is like in 24% of treated patients. And this is of course something that you have to then again treat against. And then you give, for example, steroids to block the immune system, or anti-TNF antibodies to tone down the immune response. Yeah. So nothing is without its side effect, and therefore it's very difficult at the moment to judge how much needs to be done. Yes, Alex.
Do you know how long these side effects stay? Because when the antibody is gone, like after 21 days, half time, it should be reduced afterwards, or?
Yeah, so the half-life of these antibodies is 21 to 23 days. So I would say that, yeah, you give high amounts of these antibodies, so it will not be completely gone after 23 days, but maybe after 100 days it will be done. Yeah, doesn't matter because the patient will not survive that long. So they get these off-target toxicities, and then they get treated within days usually, because otherwise they would die. Yeah.
Okay.
So you start this initial immune response, but at some point you have to stop it. It's not like you wait until this goes away by itself. If you have this high degree of T cell activation or B cell activation, then you have to stop it.
Okay, thank you.
Okay. There are two other drugs that target PD-1. One is called pembrolizumab. The other one is called nivolumab, by different companies. All are intended to treat patients that would die otherwise, like that have advanced melanomas or skin cancer that has spread to the whole body, or non-small cell lung cancer that is also very difficult to treat, or metastatic renal carcinoma. The PD-1, the anti-PD-1 antibodies seem to work a little bit better. So the response rates are like 10 to 50%. So it means 10 to 50% of treated patients will do much better with these antibodies, and only 10% of patients develop these adverse effects. So yeah, this has led to a lot of clinical studies. So these are numbers from 2020, where there were just 334 studies going on. And in 2024, it's already 1,350 studies going on. So there is an intensive interest in further developing these checkpoint inhibitors. But these drugs have challenges, as Alex already alluded to. So we don't know, do we actually need these strong immune responses? Are only the tumors sufficiently killed if you have these strong immune responses? Or can you directly give them with, for example, some steroids and it still works? Then the question is, how can they be safely combined with existing and novel treatments? For example, with, I don't know, the cancer vaccines or the CAR T cells that we will look at in a minute. How strong of an immune response do we need? And how long should we treat? That's exactly Alex's question. So this is just things we don't know, and we have to try. And at the moment, we are learning the hard way. We were learning this by using this in patients which have no other choice. Yeah. Okay. Then we will round up today's lecture with the last strategy that is even more, I think, promising. It's the CAR T cell therapy. CAR is not a car that you drive around with. It's a chimeric antigen receptor. What is this? So you, in this strategy, use so-called CAR T cells. And if we look at this more closer, so if you have a T cell that is specific for a tumor, and in order for the T cell to kill the tumor, you need to activate it. And this is done in immunology by the T cell recognizing a peptide specifically via its T cell receptor. This transmits then an activating signal into the nucleus of the T cell, telling it to become activated, but this is rather weak in terms of signal. And you need this peptide doesn't come from nowhere, it's not swimming around it, it's presented on the surface of an antigen presenting cell that has taken up the tumor antigen, processed it into a peptide, and presents it via MHC class 2. So they need two cells, basically. And then you have the antigen presenting cell also providing co-stimulatory molecules, which then give a much stronger activating signal into the nucleus of that T cell. And only if all these things and signals and cells and stuff comes together, you can get an activation of the T cell. However, there's an easier way for the immune system to be activated, because our immune system has structures that can directly recognize, for example, tumor antigen, and these are antibodies. And they even can react to peptides, but they usually react to the intact molecules. So you don't even need the whole antigen presentation for this. They can directly react to correctly folded proteins. They can directly react to also sugar structures. And what was now done is they have generated a Frankenstein receptor by fusing the top part of an antibody that recognizes the antigen with a spacer. It's usually a CD8 molecule, and intracellular parts that transmit an activating signal. And this then can directly result in the activation of a T cell. So you basically have improved the T cell by introducing a receptor that can directly activate the T cell. But again, before Alex raises her hand, you have also eliminated the control by the antigen presenting cell. Yeah, so these cells, once they see their antigen, they will start the fight. Yeah, so this can, of course, again result in problems. And the intracellular parts of this CAR cell transmit even stronger activating signals. And this would result in, if you have these CAR T cells in patients, that they efficiently recognize the tumor and can directly kill it without the help of any other cell. There are different generations of CARs. The first generation of CARs did not have the sequences inside that are needed to transmit the co-stimulatory signal. This is then for the second generation. We have these domains, as they are called, like 41BB or CD28. Or in the third generation, we have even multiple of these co-stimulatory domains. So the first generation still needed a co-stimulation from an antigen-presenting cell or from some kind of other signal. The second and the third generation of CARs are fully self-sufficient in activating the T cell. What then happens, I don't want to go too much into detail, is that once the antigen is bound on the outside, it triggers all kinds of activating signals in the T cells. If you are interested in this, you can press on pause. There are a lot of different molecules that probably will not tell you anything. Some might say something to Alex and Hannah because they are like in advanced stages of their PhD thesis. But basically what they all do is they trigger different transcription factors in the nucleus of the T cell that then allow the T cell to become fully activated and fight that cancer. Just one small clinical example. I think it's a groundbreaking study. It's a little bit old, but it was like the first one. It was a CAR directed against CD19. CD19 is a molecule that is expressed on most B cells. And it was used in patients that have acute lymphoblastic leukemia. So they have a cancer where their B cells have turned into cancer cells, and they then cause so much trouble and destruction that they kill the patients over time. And then these patients were treated with CAR T cells that were directed against CD19. And out of 30 patients in 27, of them, that's 90%, all signs of cancer disappeared. That's crazy. It's like 90% that the cancer is gone, and before CAR T cell treatment, most of them would have died. And out of these 27 patients, 19 have remained in remission, so they are basically cured from cancer. And 15 out of these 19 received no further therapy. So only 4 of them needed some other kind of treatment. And these are like response rate and treatment success rates that were unthinkable before. And now we can treat certain kinds of cancers with these cells that would have killed the patient in 100% of the cases before. How is this done technically? So you have a patient with a tumor, you isolate some T cells by, for example, taking some blood. Then you stimulate them in vitro, so in petri dishes in the laboratory. You activate them with all kinds of factors, like interleukin-2 is the one we learned about in the first strategy that activates the T cells. And then you transfect them with certain viruses. So you use viruses that infect the T cells to basically transmit the plan how to build the CAR T cell receptor into the genome, into the nucleus of that cell, and then it will start making the CAR receptor, make antigen receptor. Then you give them some rest because this is a stressful event, and some cells also die during that procedure. And then you maybe treat them very well, send them into the recreation hotel to expand them to get sufficient amounts, and then you infuse them back into the patient and they can start killing the tumor. Problem is you have basically T cells that can be activated, antigen specifically now compared to the strategy before, but they cause very strong immune responses. And then this causes diseases like cytokine release syndrome. So you will feel very sick if these cells secrete a lot of cytokines, and many patients secrete high amounts of interleukin-6. This is a strongly inflammation-inducing cytokine, and this sometimes has then to be blocked again with steroids and anti-interleukin-6 molecules, which are also available in the clinic because they are used for autoimmune diseases, for example. And then, depending on what your target is, you have to be careful, because if you treat these lymphoma patients with the anti-CD19 CAR, you have killed all their B cells. So they will be cured, but they will be immunodeficient afterwards because you have killed all their B cells. So you need then to provide them either with some new B cells, or give them some antibodies or some antibiotics to make them, again, immunocompetent, at least to a certain degree. And you can also see here, this is again the numbers for clinical studies on CAR T cells. It is, again, quite a high number of these studies showing that this probably has a lot of promise. So if we come to the end, I've brought you here like a slide I modified from a textbook. It's not important what is on the slide, but what is important is to realize that there were some breakthrough things discovered, like in the last century. Then there were some even more things discovered in the last century. And then in this century alone, in the first 25 years, we have made tremendous progress by just understanding how cancer development works, understanding how the immune system can control cancer, and we have made really tremendous progress. So that I think that many of the cancerous diseases that are a big problem, or were a big problem, will not be that much of a problem, and maybe they will turn into chronic diseases that you can live with rather normally.
The best time to have cancer is now.
Or in 10 years, probably.
Yeah, of course.
Yeah. Yeah, it's that's what Hannah's saying. So,
but of your lifetime, the best time to have cancer is now.
Yeah. So if you would have had leukemia 30 years ago, you would have died. Today, you might be cured. So if you come to the summary, cancer is a disease caused by an uncontrolled division of abnormal cells in your body. And this is then triggered by certain stimuli that turn these proto-oncogenes that normally regulate, for example, the cell cycle into oncogenes, and then the cell cycle is running uncontrollably, allowing these cells to grow uncontrollably and to spread in your body. Yeah? And this is normally controlled by the immune system with the 3E model that eliminates usually the tumors, but some tumors then escape, and they then start to modulate the immune system by, for example, inducing these regulatory T cells or suppressing T cell responses by themselves. Tumor treatment is done by a cascade of surgery followed by radiotherapy and chemotherapy, and then when the tumor load is low, we do immunotherapy to eliminate the last few tumor cells. And then we have made tremendous progress in the last 2-3 decades with inducing anti-tumor responses that discriminate between tumor cells and their normal counterparts. And here especially the therapeutic vaccines might be a big changer in future, but especially the therapeutic antibodies now already are a big game changer, and the checkpoint modulators and CAR T cells are also very, very good. With this, I would ask the two of them if they have any further questions. I know it's a difficult topic because we all know people that have had cancer or have cancer at the moment, but at least we try to give you some information that you can understand what's going on and how and why these diseases are treated in a way they are. Okay, if there are no further questions, I thank the two of them again, really, for doing this with me, because they also find a lot of times where I talk just gibberish, and then they correct me, and then we can have this in a way that is maybe good for you to understand. If you two out there have questions, just post them in the comment section. We try to get back as fast as we can. And with this, I thank you all for watching, and we see you in the next video. Bye.
Bye.
The title says Tumor Immunology.
Oh, Hannah is too smart, eh? And she can read the stuff. She has good eyes.
I think it's because we are playing so many riddle games.
Mm-hmm. Isn't our job a big riddle game? Isn't life a big riddle game?
Actually. But it's so hard to find the ways.
That we switch to English.
We have no other choice.
Okay, Alex, you need another chair at some point.
I can change my chair now if you want. Then it's no problem anymore.
Yeah, then change your chair.
Yeah, now? Okay.
Okay, I will get another tea.
It's not gonna happen. It's not gonna happen today. Everybody's running away.
No, if she...
No. Yeah.
If she's quick, then it's... she's leaving the room. Okay.
Go! Go your freedom to get a tea.
We use the phrasal verb go off to mean that the sound is beginning to go. Okay. It shows that a sudden and spontaneous noise had started. Ah, okay. I'm sorry, I was not aware of this.
English learning with Alex. Take 2.
You never said this in the last 8 years,
yeah,
so it's your fault.
Because I was talking English. Okay, again, then there is the second checkpoint. It's at the end of the G0 phase, so the cell has then decided...
Again G0.
G0? Oh, what is wrong with me? What is wrong with me? I'm a G0 guy, obviously. All of immunology happens in G0 phase, so that's...
Yeah, but all the cancer stuff is happening between S and G1.
Okay.
Or not all, but...
Most. But it's okay so far? Okay. Cool. Okay. Yeah, Alex?
I have internet problems.
But actually, Hannah's controlling me, yeah? Okay.
Yeah, I think I will just turn off the computer again and come in again.
Okay.
I hope
I will
this
just
would fix it.
continue, yeah?
Yeah, or you make a pee-pee break.
Not yet.
Not yet, okay. Then see you soon, hopefully.
See you soon. That can be used to attack the pathogen or to instruct the immune system to attack. Now that can be used. Not the pathogen. It's a tumor. It's a tumor. It's a trap.
Attack the tumor.
It's a trap.
Yeah, actually it is.
Yeah, Alex, you can clap. Alex has graduated successfully from clapping university. Okay.
Doctor degree in angel studies.
Angel studies. That sounds interesting.